All right, very good. So we're still in chapter 33. We'll spend actually a couple more lectures in chapter 33. There's a lot to talk about in this chapter, right? The vertebrates are a very big group. What we said the other day, about 95% of all the animals out there are invertebrates. So we got quite a few, 5% of the animals to go through, and we've been waiting organisms that animals, but still kind of playing with it. And I don't see that as we all a lot about what connection bad how. I mean, what's happening. Sound bad video. Let me turn the video off. What happens? Good. Yeah. Sound better now? Does it sound better now, folks? Yeah. All right. Just let me know. Keep me praised if it gets any worse. I've turned the video off. That might help with some of the streaming a little bit. So again, when we're talking about the sponges, we're dealing with organisms that are considered animals. They're considered multicellular, but again, not quite tissues, right? We don't have tissues. We don't have organs in sponges. We do have these very interesting little cells here. Oh, I forgot my pen at home, and I'm going to end up. Oh, I'll use this one. These very interesting cells. Why won't this work? There we go. These right here. These choanocytes. Remember, choanocytes look an awful lot like choanoflagellates, which were the protozoans. So that's a really neat bridge between the protozoans, right, and between the animals. We can see a very clear a connection here between a group of choanoflagellates of unicellular protozoan that were cooperatively living together, and as you kind of advance that in evolution, what we get is an organism like this, a sponge that's sort of taken those choanoflagellates and that multicellularity of them and kind of turned them into an organism, right? But still, not quite an organism that has organs and tissues yet. We move on to the next group, and again, let me remind you that this part up here where it says diploblastic should actually only include these two organisms. The ctenophrans and the cnidarians are the only ones that are diploblastic. That's really sort of misleading the way it's written with the bracket up here. Sponges aren't diploblastic. Remember, diploblastic means you have two tissue layers. You have two tissue layers. Some organisms have three tissue layers. And the tissue layer I'm talking about, remember, is the embryonic tissue layer. So it's the ectoderm and the endoderm and the mesoderm. So I'm not talking about the tissue layers that you end up with. I'm talking, oh, it's a Gulf fritillary butterfly. How cool. I'm talking about the, this is why I could never sit next to the window when I was in high school, right? I'm talking about the germinal tissue layers, the embryonic tissue layers. And ctenophrans and cnidarians only have the ectodermis and the endodermis. They are ectoderm and endoderm. They don't have the mesoderm, the middle part. So we went through the ctenophrans. We looked at the comb jellies. Really cool group. Used to be jellyfish, or used to be lumped in with the jellyfish. And again, that kind of goes back to the beginning of the semester. The idea that organisms that look alike very often got lumped together, but we know for a fact through all of the exceptions we've come across that isn't always a hard and fast rule. You have to be careful using that. The specific thing that separates the ctenophrans from the rest of the jellyfish, from the true jellies, if you want to think of it that way, is that all members of the group Cnidaria, the next group, all of them have those stinging cells, that cnidocyte. That's where the name of the phylum comes from. That is one distinguishing characteristic that all organisms in that group have. And ctenophrans, while they have tentacles, they don't have stinging cells. They have a way of capturing their prey. They don't sting their prey with the nematocyst and secrete a little toxin and do it that way. What they do is they use adhesive, right? So their tentacles are covered with things called coloblasts. And coloblasts discharge this adhesive. They trap their prey in this glue basically. And, you know, although these organisms are very similar, the way they came about in terms of prey capture indicates that they probably have very different evolutionary origins. And that's why the phylogeny down there is unclear of the comb jellies at this point. Now, when it comes to the true jellies, like I said, the distinct characteristic of all true jellies is the fact that they have a special cell called a cnidocyte right down there, and that's what makes them cnidarians. Plane flying over in the background. I guess I didn't consider that when I was sitting outside, that I'm next to the airport here. All right, so that's why the comb jellies aren't in this group. They don't have the cnidocytes. Okay, very simple body plan with these organisms. Basically, they have an ectodermis, an endodermis, and the endodermis in this case is specifically called the gastrodermis. That's still endodermal tissue in the embryonic stage, and that became the gastrodermis, which is the digestive part. These organisms, unfortunately, as we learned in class the other day, only have one way in and one way out of their digestive system. So everything comes into the mouth, and all the waste products are then dumped out through the mouth as well. Also remember, this group has two different forms, the cnidarians. You can be the polyp or the sessile form, or you can be the mobile form, the medusa form. And oftentimes they will switch between these forms, or in some species, they're only one form or the other. We looked at Obelia. This is actually where we left off on the previous slide. So this, we looked at Obelia under the microscope. We looked at Obelia in the lab. I said there are two types of polyps that you need to focus on. You need to focus on the feeding polyp, which has got the tentacles, and then the reproductive polyp. Where are we? Right here. The reproductive polyps. All right, and the reproductive polyps, they give rise to medusa. See, after the reproductive polyp comes the medusa. This is the Obelia, is a sessile stage, okay? So out of there comes the medusa. Here's some little medusa Obelia, by the way. Right, so these are with the tentacles facing down, and this is how reproduction occurs. So I said the other day in class that Obelia, the reproductive polyps, little medusa, are good for dispersal. But involved in that idea of dispersal is when these medusa are sent out into the environment, or when they're released, they then engage in sexual reproduction. So the medusas have the gonads, they produce the egg and the sperm, fertilization occurs, and we don't have the alternation of generation like we did before, and we form that zygote. That zygote eventually becomes a larval stage known as a planula, which really doesn't look like much. That's the planula of a cnidarian, that larval stage. That settles down and develops back into a polyp again. So Obelia, like Hydra, these two up here, the Hydra that we looked at, are polyp forms of cnidarians, and in the case of Obelia, it's what we call a colonial cnidarian because it lives in this colony of reproductive and feeding polyps, and it uses the reproductive polyps to reproduce as well as to disperse. Many cnidarians can reproduce through asexual reproduction. They do have a male and a female. Now, the word you see there, gonorchism, up here, indicating male and female. I'm trying to draw with my finger. Can I do that? Right. You see up here at the top where it says gonorchism. Oh, scribbled the wrong thing. Gonorchism means males and females. The word dioecious that we learned back in plants. We were talking about cycads, for example. We said cycads were dioecious. Remember, dioecious is males and females. The word gonorchism is a term. Dioecious and gonorchism are synonyms, basically. We generally leave gonorchism as the term we use to refer to animals that are males, that have a male and a female. So, unless sounds like probably confused the heck out of you guys, but dioecious. So animals can be dioecious, but typically the word gonorchism is a dioecious animal that we're talking about. So in other words, you wouldn't use gonorchism to refer to the cycads. So the zygote, the larvae, the polyps, right? It's all recycled. Coral species are a polyp with the exoskeleton of calcium carbonate. What coral species do is they secrete that calcium carbonate, and that makes the outside skeleton of them. That's what gives the reef how hard it is, the toughness of the reef, and it's good for protection. The major evolutionary advantage, a major evolutionary innovation that occurred in these organisms is we have extracellular digestion. Now that's the same thing that the fungus had. Fungus had extracellular digestion, but this was extracellular digestion on the inside. okay, as opposed to the fungus, which the extracellular digestion was on the outside, recall. So the big thing here is extracellular digestion occurs on the inside in the gastrovascular cavity, and the food fragments are then taken up into the cell phagocytosis. Now, if you think about it, the biggest advantage you have over extracellular digestion occurring on the inside is less loss of food, less loss of nutrients. If you're doing the extracellular digestion on the outside, then some of the digestion or some of the materials you digest are going to be lost into the environment. So you, the organism, you don't get access to them. But if you're doing that digestion in that gastrovascular cavity, right, in that space on the inside there, then you're going to lose a lot less food. So it's much more efficient, much more efficient. That's one of the major evolutionary innovations of the members of this group. Okay, let's look at different groups or clades of cnidarians. First of all, let's look at the Medusozoa. I don't know why that's highlighted in red and made bigger than the other. It just is. It just was a mistake in formatting. There's nothing significant about that. Medusozoa include these groups down below: the Cubozoa, the Hydrozoa, and the Scyphozoa. And all Medusozoa clade produce a medusa, the part with the tentacles facing down. And they include the really cool the box jellies. Chironex fleckeri in the upper right is a box jelly. It's a box jelly. Very strong swimmers. Stings of this organism can be fatal to human beings. More venomous than a cobra, by the way. We don't have any venomous box jellies around here in Florida. Most of those occur down around Australia, which, you know, once again is where you're going to get all those strange animals. The Hydrozoa are the Hydra, which you saw, and then another member of this group is the Portuguese man of war, which is actually what we call, more specifically, something called a siphonophore. Portuguese man of war, man, they're a really weird critter. Portuguese man of war are a colony of cells. Again, it's what we call a colonial organism. So it's not quite the same as a multicellular critter. This is, again, one of those critters that seems to have a foot in the old world of cells aggregating together but not quite being tissues. So, for example, in the Portuguese man of war, there's one set of colonial cells that makes the sail, the thing at the top. There's another set that makes the actual, body part of the organism, and then there's another set of cells that makes the tentacles. And each of those cells live independent of the other types of cells, but all together they live cooperatively in that organism. So kind of cool. A Portuguese man of war is really sort of three critters living together, three types of critters living together. Hydrozoas have, in many cases, a polyp and a medusa stage. That would be the Obelia. Hydra, though, is an exception. Hydra only has the polyp stage. Remember, so not all of them have medulla. Freshwater, we talked about this in class. Hydras are the only ones that, Hydrozoas and Hydras specifically, the only ones that are freshwater cnidarians. Now, the ones you're most familiar with are the Scyphozoa, the jellyfish. They live most of their life as a medusa. If you've, ever been anywhere in Florida in the water, you've probably seen, obviously jellyfish. There's a jellyfish. This is an upside down jellyfish. Lives down in the Florida Keys. It's called Cassiopeia. Something like that. Cassiopeia. It's an upside down jellyfish with the tentacles. It's not polyp. It's not attached to the bottom. The one down below is a new group of cnidarians that are basically sort of a mixture of a polyp and a medusa. They're kind of medusa at the one end up here, and they're also attached at the other end. So they kind of have both worlds. It's not or medusa or polyp at different times. It's medusa and polyp together. Again, all these critters have stinging cells, even the little Hydra. Now, the other group, I guess maybe now why I put those in red is maybe because I'm separating the two groups of cnidarians. So the medusa are the medusa ones that make the the tentacles facing down. The anthozoa tend to be the polyps. Those are the sea anemones, the corals. Beautiful purple sea fans. I've got a couple of those in class. I'll show you. Those are neat. What kind of feeding do you think purple sea fans engage in? How do you think they get their food? Any ideas? Yeah, other organisms, but specifically, how do they get access to those other organisms? By looking at the way their structure is there in the picture. What does it look like? Filter feeders. Yeah, filter feeders, right? They have all those little open spaces and the water flowing through, and then they filter out the food that they want. Members of the anthozoa group, the sea anemones and the corals and the sea fans, only have the polyp form, and they can either live solitary or colonial. That's what corals are. Like that brain coral you see down at the bottom, the healthy and the bleached. You see why it's called brain coral, right? It looks like a brain. Yeah. A brain coral or corals are a group of individual organisms living in a colony together. Now, the corals down below, we only have in the United States one continental coral reef. In the continental United States, I should say. I kind of said that backwards. There's only one coral reef in the continental United States, and that's the one that's down in the Florida Keys. The other one is, Hawaii, but that's not the continental United States. Unfortunately, with the closeness and proximity to human beings and all of the things that come off of up the water and go into the ocean, the corals down in the Keys, many oftentimes are taking a beating. And you can see, if you look at Caribbean coral reefs in general, the percent of coral cover has dropped considerably since 1970. And that's directly related to human factors, global climate change. Corals are strange organisms. They need to live in shallow water because inside of corals there are these symbiotic dinoflagellates called zooanthellae. In the picture here, the zooanthellae are the little brown dots that you see scattered throughout the tentacles of that organism. Those are dinoflagellates, protozoans, right? Symbiotic dinoflagellates. And what they do is they photosynthesize. So it's a mutualistic relationship because the zooanthellae provide food when they photosynthesize for the coral, and the coral provides not only a place for the zooanthellae to live, but they also provide carbon to the zooanthellae. All living things need a source of carbon to build their molecules from, so it's a plus-plus relationship. However, here's what happens. Whenever corals feel stressed, right, whenever finals are coming due and they got bills to pay, right, whenever they feel stressed, now corals don't get stressed by that, get stressed by temperature changes, waters. Corals are very much like Goldilocks. They got to have it right in the middle, not too hot, not too cold, can't be too salty, can't be too not salty. It's got to be right in that middle range. But since zooanthellae are photosynthetic, corals are usually going to be found in shallower waters, because the light doesn't penetrate very, very far down into the ocean. So they live in shallow waters for the photosynthetic part, but living in shallow water means you're closer to land, and closer to land means more pollutants and nutrients and all kinds of gunk, things that could stress corals, flow off of the land. And in shallower water gets warms up faster, cools off faster. If there's been a lot of rain lately, then that shallow water area, the salinity will drop a little. If there hasn't been a lot of rain, then the evaporation might drive the salinity up a little bit. And corals are very sensitive. That's why they're hard to keep in aquariums. You've ever known somebody who tried to keep corals alive in an aquarium. Well, anyway, when corals get stressed, they do this weird thing. They actually expel, they actually kick out or get rid of their zooanthellae. Now, this is a death sentence. Down below, you can see the healthy coral on the left. The coloration in coral comes from the zooanthellae. It's not the critter. And then on the right is the same head of coral, same brain coral, but it's now been bleached. Bleached means that coral animal, those coral animals, that's colonial, have gotten rid of all their zooanthellae for whatever reason. And when you get rid of the zooanthellae, obviously you lose the benefit of the photosynthesis. So like I said, it's actually a death sentence to do that. Now, a lot of corals, or sometimes, corals can reincorporate the zooanthellae, so that bleached coral on the right can take the zooanthellae back up and everything's fine. But a lot of times that does not happen. A lot of times when the coral bleaches, when the zooanthellae are expelled, the coral head dies. And that's why we're getting that diminishing in the coral cover in the Caribbean. We're getting these zooanthellae that are being expelled with all the stuff. You think about the Caribbean, all the people living in there and all the stuff that flows into the water near shore especially. I know when I was down in the Keys looking at coral reef health, we could tell when there had been a really heavy rainstorm up around Miami, toward the tip of the Florida Peninsula, because all that rainwater a couple days later, we'd be out over the reef and we could detect it because the salinity out over the reef would drop. Because rainwater is fresh water. Salinity is a measurement of the salt in the water. So when you add all that fresh water, then you're going to drive the salinity down. And that was down off the Keys based on rain that occurred on the mainland, miles up into the state. So all of that stuff that flows down through the state eventually makes its way out over the Florida Bay and then out over the reef. So that's why we have such an issue with that. If you have never had a chance to get down there and do some snorkeling or do some scuba diving down there, you should definitely try that. It's incredible. All right, where are we now? Oh, now we're going to move on into the triploblastic critters, and we're going to look at those that are bilaterally symmetrical. All right, so the ctenophorans, the cnidarians, they had radial symmetry. Most of the rest of the critters you see have bilateral symmetry, even though I did make a mention in class and I said that the echinoderms, which we'll talk about later on, not today, sea stars and all that, they actually have both kinds of symmetry. Their larva is bilateral, and as adults, they have radial symmetry. Okay, so at about 670 million years ago, we have this branching off and this diversification of the bilateral organisms. Most of them having a complete digestive system. Most, not all. So again, in the complete digestive system, you get the mouth and the anus. Remember, before that with the cnidarians, we didn't have that. So we're going to take a look at all of these different groups. Obviously not today. We won't get to all of them, but we'll start with one that we looked at in the lab the other day. We'll start with the flatworms. All right, now a little bit of word root terminology here. Platy means flat. Platypus has a flat bill, right? And helminthes means worm. You go to the doctor, you go to the vet, and you or your pet has worms, your doctor will oftentimes prescribe something called an anti-helminthic, and that's a medication to get rid of the worms. Now, years ago, we used to lump all the worms together into one big group and call, I can't remember, it was something like vermiformes or something like that, some old name. But that was based on that misinformation that we get, that just because an organism looks alike doesn't mean that it's related. So we looked everything that looked like worms, like the flatworms, the roundworms, the segmented worms, all of those were lumped together. You can see quite clearly, for example, even in the roundworms are in a totally different separate group. So platyhelminthes, that word literally means flat worm, and you saw them in class the other day in the lab, and indeed they looked like a flat worm. There we go. Ciliated, soft-bodied, found everywhere. Marine, moist terrestrial environments. I mentioned to you that we do have flatworms. We do have a flatworm here in Florida. I think I have a picture of it. Yep, I do. I'll show you one in just a second, a picture of the flatworms we have here in Florida. Acoelomates, no body cavity. Now, this is an exception. They still have an incomplete digestive cavity. So that pharynx that you guys were looking at, you can see it in the picture here. There's the pharynx. That pharynx is a muscular structure that masticates. What does it mean to masticate food? Chew. There you go. So that muscular pharynx mashes up their food. Many species of platyhelminthes are parasitic. Now I want to ask you, why are they flat? Why do you think platyhelminthes are flat? Now they're going to stay small. I might give you a little bit of a hint. But what's the advantage of being flat? Now remember, these organisms, they don't have a circulatory system. They have a gastrovascular system. So they don't have a circulatory system like us. They don't have gills. They don't have lungs. I think I've given you guys enough information now. Why are they flat? Anybody know? Do you remember why mosses had to stay small, had to stay low to the ground? I mean, what was the big, more efficient, but more efficient in what regard, Thomas? Oh, yeah, there you go, Nicole. Take a look at this. Back to that old surface area to volume ratio thing again, right? Now, don't worry about the numbers here, the calculations, but just look at the ratios. Remember, we said that cells stay small because the higher surface area to volume ratio is more efficient. So if we take a cube of similar size, smaller cubes, and we arrange them in a cube like that, you have a 2-to-1 surface area to volume ratio. Okay. But if you take the same number of cubes, same size, and you lay them out flat, you have a 3-to-1 surface area to volume ratio. The higher the surface area to volume ratio, the more efficient, right? Higher surface area to volume ratio means that you can pass things across the membrane a lot easier, back and forth. Water, food, nutrients, waste products. And if you don't have if you don't have lungs or gills or some way to get rid of those waste products, you're limited to diffusion and osmosis, right? So flatworms stay small. There's a flatworm in the middle. That's a cool looking one, isn't it? They're going to stay small and they're going to stay flat because that's beneficial. Surface area to volume ratio is a common topic in biology, not just with platyhelminthes, not just with flatworms, but you remember in the thylakoid of a chloroplast, if you remember in the thylakoid of a chloroplast, there are all those granum. And on the granum, on the membrane of the granum is where photosynthesis happens. And you remember there were lots of those granums stacked on top of one another. Or maybe if you remember from the mitochondria, that inner membrane was wound back and forth where all that oxidative phosphorylation occurs. So what you're doing is increasing surface area. In the case of photosynthesis and respiration, the surface area that you're increasing is the membrane by which those reactions happen on. Remember, you got to have a membrane in order to build a gradient across it, which is what you do in photosynthesis and respiration. So the more membrane, the more of those proton pumps that you can have. So you see that a lot in organisms. We saw it when we looked at the fungi. That's a mycelial mat. We talked about the fact of how much that increases surface area. And this would be an artist drawing of the villi, the little projections off the cells that line your intestines. So as food is traveling down your intestines, there's a lot of surface area to absorb the food. So see, this whole thing of surface area to volume ratio, you know, it's repeated many times throughout living organisms. It's a very effective way to make that organism much more capable of moving things across its membrane if you had a lot of surface area per unit of volume. So it doesn't apply to just cells staying small. Okay, the two clades of platyhelminthes. The first one is the one more time, Caneluda. It's Caneluda. No, hard time this morning. The other one I could say Rhabdophora. One down below, Rhabdophora. Catenaluda. There we go. Catenaluda is a small group of about 100 species. They're called chain worms. This is what they look like. They look like a chain, don't they? Typically, they reproduce by budding, asexual reproduction. The offspring budding before they actually detach. The one down below, this is the one that you find in Florida. This is the flatworm that we have around here. About 20,000 species of this group. Free living species. Most of these kinds of organisms, this one here looks like the one we saw in lab, the planarian. Most of those are free living. There are some parasitic, like this one here. You know what that critter is on the right? Yep, that's right. That's a tapeworm. Trying to see if I... There we go. Okay. The free-living planarians, like, for example, the one we looked at in class, the Dugezia. No circulatory system, diffusion and osmosis. That's why they're small, right? Digestion, some of the digestion occurs out of the body, and they can absorb some through their body walls. They're thin and small enough. Some of it occurs in the gastrovascular cavity, and then the gastrovascular cavity is what moves the nutrients throughout the rest of the body. So it acts as kind of like the circulatory system in us, right? Parasitic species lack a digestive system. If you're a parasite, oftentimes you don't need a digestive system. For example, tapeworms, like in that previous picture, tapeworms are nothing more than reproductive parts. It's a head that connects at the front, and then it's just a bunch of reproductive parts after that. And tapeworms, since they live inside of the host, they're endoparasites, they don't need a digestive system. They just absorb the material from whatever host they're living inside in the digestive system. So if they're living down in the intestines, right, and all that food is coming by and it's already digested and it's liquefied, all they really have to do is absorb it. It's kind of like ABC food, right? Already been chewed. Very simple nervous system. They have what we call an anterior cerebral ganglion. Now, a ganglion, I put in quotes, a ganglion is defined as a collection of nervous tissue. You have ganglia throughout your body, but a massive collection of nervous tissue, in your particular case, would be up at the brain. So in the case of the planarian, the anterior ganglia are its brain. It's not really a brain by technical term yet, but nonetheless, it is a collection of nervous tissue up near the front of the body, right? We talked about cephalization, how important that is. Put all those sensory organs in the front because that's the part of the body that comes into contact with the world first. An eye spot, right? Just light and dark. This is one of the, we talked about how eyes evolved. This is just a very simple senses, light and dark. So not an eyeball, right, Jennifer, but an eye spot. Reproduction, most oftentimes, though, even though you're hermaphrodites, you're going to fertilize another hermaphrodite simply because you want to remove the inbreeding issue. They can reproduce then sexually. They can also reproduce through fission, that you can see on the right. They can actually split off and form another worm. You can actually cut them too. You cut in the laboratory, you cut them in half, and they'll form another worm. So it's kind of like a little bit like the sponge, right? The fragmentation. Okay, now they're excretory or they're osmoregulatory systems. An excretory system is, well, it's exactly what it says. It's how nitrogenous waste and metabolic waste and other kinds of things are deposited on the outside of the body. The osmoregulatory system is like the contract vacuole in the paramecium that we were talking about, right? That's what maintains water balance. So how do you keep the right amount of water? You don't want to be in a hypotonic or hypertonic environment. You don't want to absorb too much water or lose water. So its excretory and osmoregulatory system is controlled by these things called protonephridia. Now, that word might have a root to it that looks familiar. Let's see if I can pull it out. What is nephron? If you went to a doctor, you might go to a nephrologist. But you ever had to go to a nephrologist? I think I spelled that right. What does a nephrologist study? I'm going to see if I spelled that right. No, I spelled it right. Oh, there's one right down the road here. Did I look it up online? Nobody knows what a nephrologist does? Nobody wants to go to med school and become a nephrologist? Kidney. There you go, Nicole. Yeah. So a protonephridia is proto meaning initial or first. A protonephridia is a very basic, very simple type of kidney. A very simple type of thing that does the job that a kidney does, which is getting rid of these waste products. And it's made up of this network of little tiny tubes that are found throughout the flatworm's body. And those tubes are capped at the end with these things called flame cells. Now, the next picture, I'll show you a little more detail what a flame cell looks like and how it works. But that's how it gets rid of nitrogenous waste, excess water, so liquid kind of waste. Metabolic waste, larger things, are excreted into the gut and then eliminated through the mouth. Remember, these guys don't have an anus. Flame cells got their name. Now, let me go ahead and show you the picture. There we go. These are flame cells. Flame cells got their name because inside the flame cell there's this massive cilia. And that cilia, I'm going to turn the camera on. I want to show you. And what that cilia is doing is it's waving back and forth. And of course, you know what that's doing. That's moving water. Just like a filter feed or anything else. It's moving water and it's gathering water into these tubes And then eventually those tubes will exit out through an opening in the body wall. The reason why they're called flame cells is because they look like the flickering flame on a candle. That little cilia, that red part in the picture, as it's flicking back and forth, I guess reminded folks of it looks like a candle. So we call them flame cells. But it's the same basic principle that we've seen in the filter feeders and everything else. If you got the cilia moving back and forth and you're moving water, in this particular case, you're bringing in all that interstitial fluid. That's all the fluid in the body moving into those flame bulbs or those flame cells, and then eventually being exported outside. So very efficient, very good at getting rid of metabolic waste and of course excess water as well. All right, folks, any questions on that? All right, one of my favorite topics now too, parasites. I parasites fascinating in maybe a macabre sense. About half of the Rhaptidophorans, about half of those are parasitic. And those are broken down into two major groups. We call the trematodes, and then, of course, as we saw in the previous picture, the tapeworms. The trematodes are also known as the flukes. And trematodes will, being parasitic, oftentimes have suckers and anchors and hooks, things to hold on to its host. And many flukes have a very complex developmental stage. They may oftentimes have multiple hosts, like you see the one on the right, the blood fluke. About 200 million people a year are infected by blood flukes. Blood flukes are very often found in the water around rice paddies. Rice is grown in standing water. And folks walking around there tending the crops would presumably maybe not have shoes on. And any little cut, a little opening in the skin, these worms can actually get into. And they can survive inside the human host for quite a long time. You see about 40 years. There's a male and a female of that blood fluke. But what happens if you follow the cycle, the ciliated larva comes from the fecal material that the human host would drop. And in areas of the world where rice is grown, if fertilizer is in short supply, sometimes that's what the fertilizer is. The rice paddy field is also the restroom. So the ciliated larva then will take up into a snail host first, develop into a motile larvae, and that motile larvae will then enter into the human. So in this case, it's got two hosts. I've seen some species that actually have three hosts. I did studied one in South Carolina that its eventual host was mosquito fish, but it also used ducks and snails as part of the group. So it went from the mosquito fish to the snail, from the snail to the duck, and from the duck back to the other way around, back to the mosquito fish. So it was the duck, mosquito fish, and then they developed to two hosts. Two big types of tapeworm is Taenia saginata and Taenia solium. That's the beef and the pork tapeworm. These are unfortunately frequent human parasites, especially if you're eating improperly prepared meat or uninspected meat. Now, I got a model I'll show you in lab of a tapeworm, but basically, like I said, a tapeworm is nothing but a head, or that scolex that you see down there. Look at the magnified picture of the skull. That's a pretty mean-looking sucker, isn't it, right? It's got hooks on it. It's got suckers. This is what it holds on to the intestinal wall of the host with. And all of those segments behind it are called proglottids. Now, as I said, tapeworms have no digestive system. The only thing they have is a head, and it's not really a head because there's no brain or anything like that up there. But all they have is that scolex, which is the attachment, and then they grow these proglottids in sections, one after the other behind them. Tons and tons of proglottids. And each of those proglottids is a reproductive structure. Now you look on the right, the dog tapeworm, that one, see it's stained, the mature proglottid. Notice what's being labeled there. It's got a testes and a uterus. Okay, so the sperm duct, or the sperm from one proglottid, can fertilize another proglottid, the eggs in that proglottid, and then you have all of these tapeworm eggs. And each of those proglottids then can break off and spread those eggs out through the environment. I've seen catworms sometimes that have bad infestations of tapeworms, and it'll get so bad that the tapeworm actually is growing out the back of the cat's, out of the cat's anus. Years ago, they used to sell tapeworm eggs as a weight loss remedy. Back around the early part of the 20th century, you bought tapeworm eggs, and they don't consume enough food for you to lose weight, by the way. Yeah, no kidding. But people would eat the eggs of the tapeworm, and of course, as I said, they don't consume enough for you to lose weight. And the hard part is getting rid of them. And not only that, as that tapeworm moves around in your intestinal wall with all those hooks, it tears up the tissue inside your intestines. So I think there are easier ways to lose weight. And like I said, it's not effective for that at all. But yeah, I was looking at some old magazine one time at UCF years ago, one of those old that they keep archives of old magazines, and in the back where all the selling and all the ads were, they were actually selling weight loss pills, natural. It was actually tapeworm proglottids. So you would eat a proglottid, and then the tapeworm would hatch, and you would a mature proglottid, and the tapeworm would hatch, and you would now have a tapeworm. What price for vanity, huh? All right. Platyhelm nymphes. Oh, the rotifers. We looked at rotifers in class. Oh, wow. Rotifers have a little trick here. Okay, rotifers. Pseudocoelomates, all right? It looks like protozoan. You see another, but it is true. All right, it's got complete digestive elementary. It's got corona, that circle of cilia up near the top, and they're called wheel animals because under a microscope, when those cilia are moving, it almost looks like it's spinning around up there at the top. It's obviously not really spinning around. It's food gathering, right? That's what it's doing. You can see down here in the picture, there's the crown of the cilia, the mouth and the corona, the jaws, the stomach. I mean, so this is an organism that has organs. I mean, this is a fairly complex organism, even though it's, it's protozoan-like in terms of size. Remember the other day that we had the corona, the head, the trunk, and then what we called the foot that we looked at in lab? Okay, now, there's something really neat about rotifers, and it's not just confined to rotifers. Let me tell you a story first. Up at the Shedd Aquarium in Chicago, they had a female sand tiger shark. Female sand tiger shark had been in the exhibit there for 10 years, maybe longer, 15 years, by herself. In other words, not by herself in terms of other sharks, but she was the only sand tiger shark in the group. There was no other sand tiger shark. There was no other male sand tiger shark. Well, after a weekend, some of the aquarists went into the exhibit there, and they noticed that there was the sand tiger female that was there and some sand tiger pups. She had given birth. How the hell did she give birth? There's no male. She hadn't been around a male for at least 10 or 15 years. So how did she do that? Well, the process by which that occurred had been documented in some species, rotifers, for example, and some other relatively simple organisms. The process that I'm talking about is called parthenogenesis. And parthenogenesis says that eggs can develop without being fertilized. So that's what was going on with the sand tiger. Those pups were born because she fertilized her own eggs. Now, this can happen in fish, amphibians, reptiles, some birds, not documented in mammals yet naturally. So it's possible for mammals to do this, but it's not something that we've seen naturally. So in the laboratory, we've been able to do that. Lizards down below, these species of lizards, those are parthenogenic. In other words, in some species of lizards, there are no males at all. Males don't exist. This is a bonnet head shark, similar to a hammerhead in the same group, but they've been documented to be able to undergo parthenogenic reproduction as well. So what good are males, huh? Males basically are just nothing more than sperm donors, aren't they? Some species have actually got to the point where they don't need males. It's kind of interesting, right? Think about that. If I don't have males around, I can fertilize my own eggs. How clever. Now, the Deloid rotifers, the B is silent there, about 360 species. All of those are parthenogenic, and they've been that way for 100 million years. So all of them are parthenogenic. All are female, no males, and yet for 100 million years they've survived, and the genetic variation to diversify into 360 different species, where did that all come from? Where did that, if they're not reproducing, and one of the best ways to create variation, we said, you know, the mixing of the gametes, how such that doesn't mix gametes? You guys have any idea? Variation. So what are these kinds of variation? All right, is that better? Okay, so where's the variation coming from, folks? Remember we said that fertilization is a source of variation, obviously. That's why you want to disperse, organisms disperse. We also said that you get variation from some other ways too, right? You remember the kind of variation that prokaryotes use? Right? Just those random mutations that occur in the copying of the DNA. And also in parthenogenic critters, when they're forming eggs, they're still going to be going through crossing over. So you're still going to get some variation there. So remember where we said that most variation that we see out there has to do with differential reproductivity and different organisms reproducing. In this particular case, you can still get genetic variation even if you only have one member that's procreating. Because remember, DNA, every time it's copied, every time crossing over occurs, it doesn't happen the same way every time. Even you copy a piece of DNA and you're trying to copy it faithfully, mistakes or changes get introduced. So you can still get variation even if you're not having that mixture of gametes. Now, I don't talk about the Ectoprocta or the Brachyopta. Those are the moss animals, very cool critters, but just a small group. Mostly due to time constraints. I just don't have time to go over all of those. So I do want to move now and talk about the next group in our list, which is the mollusks. Mutations, crossing over, that kind of stuff. How are you doing? Yeah, those things will lead to variation as well. Remember, there's more than one way to create variation. Big group here, 100,000 species. Only the arthropods, the insects and their relatives, are bigger. 100,000 species of mollusks. Snails, slugs, clams, octopus, squid, the chambered nautilus. There's the chambered nautilus. There's a banana slug, for reasons that are obvious. Folks, slugs are basically snails without shells. It doesn't mean, I'm not saying that the snail lost its shell. I'm just saying that the critter, in terms of morphology, a slug is just the snail. Slugs don't have snails. They can, like I said, can be with or without shells. The blue-ringed octopus from Australia, highly venomous, by the way. They can be small. They can be big. There's those big clams up to 270 kilograms. So it's like 500 pounds, man. Now, I think that whole thing of divers getting their legs caught in the clam when it closes and then it holds them on until they drown, I think that's probably a wives' tale. I'm not sure that's ever been proven. Most mollusks have stayed in the ocean. They're important to us. For one thing, we eat them, right? I love raw oysters, scallops, clam chowder, all that sort of stuff. We get pearls from them. Inside lining of a mollusk shell, right up here, that's called mother of pearl. It's that part that creates the pearl, mother of pearl, or it's also called the nacre. Let's see. Mother, that shiny region. Everybody knows how pearls are formed, right? Pearls are an irritation that gets into the mantle cavity of the mollusk, and what they do is they just try to coat it with this soft material to prevent it from irritating. Pests. They can also be pests. Oh, the zebra mussel. Zebra mussel, there's a picture of the zebra mussel on the left. It's called the zebra mussel for reasons that are pretty obvious, right? It looks like it's got black and white stripes. Zebra mussels are an exotic invasive species. Now, remember, I've talked to you about there's a difference between exotic and invasive. Exotic, it's not supposed to be here. It doesn't cause any problems. Egrets could be a good example of exotic. An invasive is an exotic that's here and is actually causing damage, like these zebra mussels. Zebra mussels grow like crazy. They foul things. What I mean by fouling things, you see that structure on the right here? I don't know exactly what that is, but that's some sort of a structure that, you know, a pump or something that's underwater. All of that fouling growth around there are zebra mussels. So they can plug up things. They're attached to the bottom of a ship. They can slow the ship down. Now, these are native from Asia, though. So how they got to the United States is sort of an interesting little bit of detective work. If you look at the map that I got there, November 2000, so a number of years ago, but in the red dots is where they spotted zebra mussels. And notice that most of them are concentrated up around the Great Lakes region. They have since, that's where they started, by the way, the infestation. They have since moved down into the Mississippi River drainage area, up in the Mississippi and the Missouri and all those other rivers that drain into it. Okay, the way we think this got to the United States, the way we're pretty sure it got to the United States, is when ships leave the Great Lakes region going towards Asia, they would be oftentimes loaded down with iron ore. That's you know, a very important material. The iron ore would be loaded on the ships, taken over to Asia, and iron ore weighs a lot. All right, so the ship is really low in the water with all this iron ore. Now, when it gets to Asia and offloads the iron ore, now the ship rises in the water. Well, have you ever been out in the middle of the ocean on a ship, and you know that if you're bobbing on top of the ocean like a cork, it's a lot rougher ride. Right? It's a lot better if you have ballast. It's a lot better if you're got some weight to hold that ship is riding low in the water. So if these ships that load up the iron ore in Asia don't have a cargo to bring back, don't have weight to put in their holds to give them some ballast, what they do is they'll use water. They fill up the holds with seawater, and that provides the ballast. So they go over to Asia. They suck in the water over there. And by the way, guess what's in the water that they suck in? Larvae of these zebra mussels. Okay. Then they come over to America through the Panama Canal, down the St. Lawrence Seaway, down into the Great Lakes region. They then expel that ballast water because they're going to replace the holds now with iron ore again. And all those larvae that they picked up over in Asia are now dumped into the water there. And since they were able to survive in that area and actually outcompete the native mollusks in that area, they have become a problem. You see these little stickers now. Spread the message, not the mussels. Kind of a cool little detective work, isn't it? How in the heck did they get here? Well, it was all about the iron ore transport that occurs. Oh, ooh. Yeah. Body plan. All mollusks have three major parts in their body. It's called a mantle, a foot, and the internal organs, which is called a visceral mass or a gut. All mollusks have that. Whether we're talking about things called chitins, look at that word again. It's not C-H-I-T-I-N, even though it's pronounced very much like that. It's a chitin. Mantle, gut, foot. Okay. The gastropods, the snails, mantle, gut, foot. The mantle, okay, the mantle is this epidermal sheet that forms the mantle cavity. It's much easier to see if we're talking about a bivalve, like oysters, and you split them open. You split that open, and you've got that gray tissue layer that you fold back. That's the mantle. The mantle covers all of that visceral mass and forms that mantle cavity. And in those who have shells, the mantle is what secretes the shell. Okay, so the bivalves have a mantle. Gastropods have a mantle. They all have a foot. They all have a gut. The cephalopods, they have arms and tentacles. Now, they don't have a foot, but that's what their foot was modified into. So the foot of a gastropod, the foot of a chitin. Bivalves have a foot. Bivalves use their foot to dig down into the substrate. That's what they do. They dig down into the dirt so they can hide, because they're filter feeders, and they use their siphons. Their siphons stick up above the water, and they filter water through them, and they get their food. What I'm showing you in the picture down below are the gills. These are the gills, and the part that's folded back right there, that's the mantle. That's the mantle tissue. So the foot can be modified as like a foot in a chitin or the snail, or it can be modified into tentacles and arms. In the squid, tentacles are the ones are longer, and usually they have two of them, and they'll have 8 arms. Tentacles are involved in reproduction, and the arms are involved in prey capture and that sort of stuff. Those organisms that live on land, like this one down here, can actually use its foot as a way to protect itself from drying out. The foot can actually seal off what's called the aperture. So this little thing down here, that's the foot. And for organisms that live on land, that's a good way to, prevent from drying out. It seals them off, so they don't have to worry about desiccation. Now, the internal body cavity, they do have a body cavity, right, a coelom, but it's been highly reduced. It's very small. The only places that the coelom is even detectable or noticeable now is around the excretory organs or around the heart, parts of the intestines. If you look in the picture in the lower left, you can see the heart. The arrow right there that I'm pointing to, see that little space? That's the coelom. That's the remnant of the body cavity. All of the digestive, the excretory, the reproductive organs, they're all in the visceral mass. In aquatic mollusks, their gills are called ctenidia. They're still gills. These are the gills. But the gills have been actually adapted for filter feeding. So they're using for extracting oxygen, right? Getting rid of carbon dioxide, same thing that fish use their gills for. But the gills have a mucus sticky substance on them. And as the water is flowing through the gills and they're oxygenating, they're also trapping food. Right? Filter feeders. Okay, another part of the mollusk that's very obvious if they have it is the shell. That's, as I said, secreted by the outer, the mantle, secreted by the mantle. Calcium carbonate. Calcium carbonate, very common material used in marine organisms. Calcium carbonate is what made up of the tests of the, the dinoflagellates, was it? Calcium carbonate was also what made the exoskeleton of the coral animals we were just talking about. Makes the shells of the shelled mollusks, protects them against predators. See this chitin up here in the rock? It'd be very hard to peel that away. Look back again at the way a chitin works. The way a chitin works is when it attaches itself down to a rock, what it does is then pulls up the middle of its foot, so it creates like a suction, like those plastic suction cups that you can get. And that means that that's holding on to that rock very tightly. Very difficult for a predator to come along and peel it off. Environment, you think about the oyster beds when the tide goes out, right? They're going to dry out if they don't have some way to close their shells, so they can work with that. Some members have lost their shells, like the slugs. Many times in that case, it's because they don't need the shell. They live on land, perhaps, or whatever their lifestyle is. The shell is unimportant. Remember, in evolution, evolution evolves innovative things, but it also, through time, organisms lose things that are no longer to an evolutionary advantage to them. I already mentioned the mother pearl. There's a really cool little graphic I found about how the pearl forms. So you got the little foreign body, little purple thing. The mother pearl is that shiny layer on the inside. The mantle is that sheet, right? That tissue that gives rise to the shell. And you can see what's happening is that the epithelial tissue of the mantle begins to grow around that foreign particle and eventually walls it off. And that's a pearl. That's what forms a pearl. You can see the picture on the right. There's a little tiny pearl. Okay, now, there's this thing called a radula. Only mollusks have a radula, and it's a very unique and really fascinating, interesting-looking feeding device. And that radula, like anything else, can be modified into a number of different things as well. But let's go to the next slide, and then I'll come back to this one, because I want to show you your typical radula. All right, here we are. Big picture, this is the front of the snail. And I want you to focus down below. Well, actually, look to the picture on the left first. There's the radula. You see it. The radula is near the mouth of these organisms. And in that picture down below, which is one through an electron microscope, the radula is made of these very hard teeth-like things, okay? And all of those muscles, those odontophore cartilage and muscles that you see there, what that does, and now you can move to the picture on the right, it creates sort of like a conveyor belt system. Now, the teeth don't move around like they would, say, on an escalator, but what it is you can see in the picture, as the radula, and the green stuff is algae. These are the scrapers, right? As the algae is picked up radula, it's passed from one of these to the next, eventually till it ends up in the mouth. That's a pretty doggone good way to be an organism that makes its living off scraping stuff off of rocks, right? Very, you're going to find these in snails. That's how snails, if you ever notice snails in an aquarium, the next time you have one of these up on the glass and you look real close at it, you can actually see in its mouth something kind of moving around like that. That is the radula. It's scraping materials off of the glass of your aquarium. Radulas can be modified. They can be modified, for example, into a harpoon. Predatory gastropods, predatory snails, can take that radula and turn it into this harpoon. Look at that. That's a pretty vicious looking harpoon, isn't it? Right? That looks like it probably will do a job. And they can then use that, in some cases, to puncture their prey. Other members can use their radula to actually drill holes into the shells of other organisms and feed upon them. Now, bivalves, which are filter feeders, clams and oysters, they don't have a radula. They're filter feeders. Now, this here, the radula, can also be modified into the beak of squid and octopus. Many of those beaks in those squid and octopus are toxic as well. So, very interesting feeding mechanism there. Only found in this group, this radula, scraping mechanism. Reproduction in mollusks, they're gonorchid, okay, so they're males and females versus hermaphroditic, mostly. Some are hermaphroditic. Some oysters can change sex. Some fish can do that. Some fish can change sex too. Oftentimes it's external fertilization, especially if you're aquatic, but gastropods, it's typically internal, gastropods being the snails, the terrestrial ones. The larval form, in the cephalopods, in the squid and the octopus, the development is direct. In other words, there isn't a larval form. A large squid comes from a small squid. It looks like a small squid. A large octopus or a small octopus grows into a larger octopus, but they look like that. But in the other groups, we have these larval forms called a trochophore, the one on the left, on the top, and then a veliger larval form. All mollusks in this group, again, other than the cephalopods, have this trochophore larva, right? This is a free-living larval stage. All of them do. Notice in the red, too, the annelids. We haven't talked about them, but those are the segmented worms. They also have the same larvae. And I've mentioned to you guys a couple times before this semester that when organisms have similar developmental stages, right? So in other words, if you find the same larvae in two different diverse groups of organisms, it often indicates a common ancestry. So the annelids, the segmented worms, and all the mollusks, the fact that they all possess this trochophore larvae indicates that these organisms have a relatively close relationship to one another in terms of phylogeny. Now, not all mollusks have that, but bivalves and marine snails, most snails are found on land, but there are some marine versions, have a second larval stage known as the veliger. Both of these, regardless of the veliger or the trochophore, now again, all have the trochophore and some have the veliger, but either way, it's good for dispersal. That's the whole point behind it. Now, mollusk, the word for just a second is aside, M-O-L-L-U-S-K, M-O-L-L-U-S-C. Either way, both spellings are correct. Okay, let's start with the four major classes of molluscans. The first one is the class Polyplacophora, literally means many plates. Poly, plaque, many plates. Polyplacophora are the chitons, and the chitons have 8 dorsal plates. 1, 2, 3, 4, 5, 6, 7, 8. 8 dorsal plates. And then they have this rim around here and that foot, and they can actually create that suction I was talking about. Now, even though it has those 8 dorsal plates, and I said segmentation is something we find very often in animals, we don't see segmentation in mollusks. So this is not an example of segmentation, these 8 dorsal plates. It's not, doesn't mean it's segmented. We're going to see, like in the segmented worms and the earthworms later on, that's true segmentation. Most of these guys are grazers, right? They're going to use that radula to scrape that food off of the rock. And again, in order to get one of these up off the rock, you got to be able to peel up off that margin and break that seal that they're holding down to the rock with. The gastropods, this is 3/4 of them. 3/4 of the gastropod, the mollusks are gastropods. This is a marine group, but there are some freshwater. These are the only terrestrial mollusks too. Of all the groups of mollusks, the only ones that have terrestrial versions, not all of them are terrestrial, all the gastropods, the snails. Most of them have a single shell, although the slugs have lost it. Things like limpets, snails, slugs. Up here, this is a limpet. This is a Florida limpet, and it's called the keyhole limpet. That little thing looks like a keyhole, that little hole at the top. That's not something that was made by another organism. Apple snail. You had some pictures of those the other day, Jennifer, didn't you? Some apple snail eggs. And then in the background are these things called nudibranchs. Yeah, they're beautiful. Nudibranch literally means naked gills. These are slugs, and they come in all kinds of different colors. And you would think with all those colors the way they are that would be easy pickings for an organism to feed upon. But in many cases, it's actually an advertisement, don't mess with me. Many nudibranchs feed upon jellyfish. And what they do is they can stand the stinging cells that the jellyfish have. But what they do is they actually take those stinging cells of the jellyfish and they don't consume them. They migrate to areas of the body and they'll collect in the nudibranch in these projections that are sticking off the top. Protects them from predators. So a predator comes down, wants to feed on a nudibranch. Oh, this would be easy. No snail, no shell or anything, right? And it gets a face full of these stinging cells found in these little projections. You can see the eyes, the tentacles. The pneumostome is a terrestrial slug. The pneumostome is for air. They take in air through their pneumostome. All right, my battery's running low, so I'm going to go over one more slide. Let me see if I'm in the place where I want to stop. I am in a good spot to, yeah, I do want to talk about this one next thing, and then we'll call it a day, because I don't want to lose this recording if my battery dies completely. Torsion. Torsion is what happens in gastropods. Take a look at the picture on the right and you'll see what torsion is all about. Torsion, you have the mouth and the anus. During development in the gastropods, what has happened is the body twists around so that the anus is actually over top of the mouth. And it creates something called fouling, right? You're basically dumping waste material over the same place you're eating. Why would organisms do this? Well, you learned a lot in evolution about it's all about compromises and balances. So if we look at that picture down at the bottom, and we imagine a beginning archetypal snail, and the shell begins to grow. Now, as that shell grows, it grows asymmetrically. We think that torsion, that twisting of the body in the shell, does a couple of things. It makes it easier to carry that asymmetrical shell. This is a wolf snail. They're called wolf snails because they eat other snails. Now, they occur in Florida. Look at the shell. It's along the main line of the body. It's easier to carry it that way. They get more efficient gill ventilation this way as well. And it also makes it easier to withdraw the body into the shell. All right, now is fouling good? No. Dumping waste material over top of where you're eating is never good. But it's a compromise. You'll take the fouling because of the way the shell is easier to move, easier to draw back into, and the much more efficient ventilation of the gills. Right? So it's all compromises. All right, folks, I'm going to go ahead and end it there for right now. If you have any questions for me, by all means. If not, you guys have a good weekend. I will see you in class next Tuesday. We'll continue on with the second part of our lab, our animal lab. All right. Let me stop the recording.